All right, now that we know all about soil, let's talk about how microbes interact with that soil. So, the first thing to think about of soil as a microbial environment is that it's competitive. So, there's a lot of what we call biotic stresses and abiotic stresses that can influence microorganisms in the microbial environment. So, biotic could be competition for nutrients, water, or other growth factors, basically the things that drive metabolism. Could be predation, so being preyed upon. Protozoa love to prey on things in the water environment. There's predation by fungi. Bacteria prey on each other. Algae can prey on things or be preyed on. There's parasitism, so parasitic relationships. And antibiotic production, so organisms that create antibiotics can actually influence other antibiotic or organisms with the antibiotics that they're producing. So that's important as well. And then abiotic stressors are going to be things like light, moisture, temperature, pH, texture, and nutrients. All of these have an impact on microorganisms, but they come from a non-living source. That's the abiotic component. So let's talk about the abiotic stresses because there are lots of them and they're going to be really important. And they tend to be the things that we can control a little bit more as environmental engineers. So light's a big one. So light's not going to penetrate below the top layers of soil. So as a result, it's going to be limiting for phototrophs throughout the system. So light can be limiting immediately for any organism that's going to do photosynthesis. Moisture is also going to be really important. All microorganisms and all organisms need water, and it's extremely important for metabolic activity. So having enough water and enough water within those pore spaces is going to be really critical to be able to do the metabolism that they need to. An interesting note here is that fungi are actually the most resistant to desiccation. So desiccation means drying out. So fungi are actually going to be the most resistant and the most resilient when we have low water concentrations. And then temperature is going to be important. Most soils are relatively resistant to fluctuations in temperature, but if we have temperature, like if we think about maybe a soil in Central America, like in Costa Rica or Nicaragua, it's going to be very different from a soil that we see in the Arctic, say in Alaska. So temperature is going to have an impact. Most of the time the soil temperature itself doesn't fluctuate, but the temperature of the soil is going to be preferential for different types of microorganisms. And here's our favorite little graph related to mesophiles, thermophiles, the different temperature ranges that different organisms like to be in. We're also going to have stresses in pH. So most soils are going to range between 6 and 8 for pH, and it's actually a very big determinant of bacterial diversity. And that's what's shown over in that graph on the right, is that diversity increases as you get towards 6, 7, and 8, and it tails off outside of that range. So it's really important to keep it in the right range. And then another one is texture. So soil is going to be a mixture of sand, silt, clay, and the mixtures themselves, as opposed to just a homogeneous sand or a homogeneous silt, are actually going to be better. When we have mixtures, they're going to be able to hold more nutrients, and they're going to allow for water and airflow in a way that something that's very homogeneous will not in a system. So we like texture and we like differences in texture. Then we also have nutrients. So one of the things that's really important is the carbon to nitrogen ratio. Carbon because carbon sources are really critical for heterotrophs. Nitrogen because nitrogen is a building block of proteins. So if C and N are low, then microorganisms in soil are going to exist in what we call either a semi-starvation or a dormant state because they're not able to reproduce and do the metabolic activities for growth. And then we also have redox potential. So we're going to have in an environment oxidation and reduction of substrates. And some of the redox reactions that we care about are oxygen to water, nitrate to nitrogen gas and dinitrogen oxide, sulfates to sulfur, and carbon dioxide to methane. These are reactions that are actually redox many times in the natural environment. And there's going to be a type of organism associated with these specific reactions. So aerobes are going to do this oxygen to water reaction. We can have facultative anaerobes or nitrate reducer bacteria doing this nitrate reduction. And then anaerobes specifically can do sulfate reduction is important, and then anaerobes that do methanogenesis is going to be important as well. So that's the abiotic stressors. So then we want to know, well, who lives in surface soils? So we've got bacteria, and they tend to be the most numerous. They don't really have a lot of branching, so there's not a lot of different subspecies associated with them. Let's kind of look down. Growth in liquid culture, yes, they're going to grow in liquids. And we don't really have fruiting bodies because they're bacteria, right? They will help us to fix nitrogen. We can see aerobic and anaerobic. They're going to be the least tolerant to moisture stretch, and they're going to have optimum pHs in somewhere in the 6 to 8 range. So I'm not going to go through this table in too much detail, but this just gives you some different characteristics about what lives in soils. So bacteria, we've talked about. Fungi, we've talked about. Acetomycetes are kind of like this weird hybrid organism in between the two. So they're kind of an intermediate between bacteria and fungi. And that's really kind of what they help to mediate some of those reactions between the two of them and interactions between the two. So let's talk about soil bacteria. So generally, if the bacteria is already there and present, we call it indigenous soil bacteria. And we normally classify them based on their growth characteristics and their affinity for carbon substrate, so metabolism, right? Normally, we're looking at diversity in terms of the number of what we call operational taxonomic units, OTUs. So each OTU theoretically represents a different bacterial population in a community. So that's one way that we measure kind of the diversity of soil bacteria. So just a note here, any methodology that's going to rely on characterizing environmental organisms via culture may actually get a very, very small subsection of the population that's not representative of the majority of the community. And we've kind of alluded to this when we've talked in lecture and talked in lab so far, but taking a soil sample from 1 meter away from the next soil sample, they could be very, very different because soils are so heterogeneous. So if we're trying to characterize the entire Earth environment, we're going to have to do quite a lot of sampling to figure out what kind of organisms predominate across a larger area. So that's really important when it comes to soil, is the fact that we have so many OTUs and different types of taxa of bacteria within one specific area. So these are some dominant culturable bacteria in soil. So if we start culturing bacteria from soil in the lab, these are some different types of organisms that we could see. And these are genus names in the organism column. So there's different species, but these are genuses. So we can have what are called arthrobacters. They're heterotrophic, aerobic, and gram variables. They could be gram positive or negative. And they're up to 40% of culturable soil bacteria, where we're actually pretty good at culturing these guys. They're really important for nutrient cycling and biodegradation. Streptomyces is a gram-positive heterotrophic bacteria. Acetomycete. And they're about 5 to 20% of culturable bacteria. So acetomycetes are a class of bacteria. And they're also important for nutrient cycling and biodegradation, but they've been important for antibiotic production as well. Pseudomonas is a gram-negative heterotroph, and they are aerobic or facultatively anaerobic, and they are 10 to 20% of culturable bacteria. They are great as a biocontrol agent. They cycle nutrients just like other soil organisms. And so that's important. And then Bacillus is a gram-positive aerobic heterotroph, and they are producers of endospores. And they, same idea, help with nutrient cycling and biodegradation. So these are some big genuses that we normally expect to see when we're culturing from soil. Most of the time we're going to see a lot of heterotrophs once we get below the surface of a soil. So these are some different heterotrophic soil bacteria that we can see. So again, these are going to be genus names for the most part in that left-hand column. So we've got acetomyces, such as the streptomyces. We have bacillus, clostridium again. So you can see whether or not they're gram-positive, gram-negative, whether aerobic or anaerobic. And you can see that there's many different functions for these organisms. So a lot of them help with carbon cycling, the production of geosmins, which actually produces what we call an earthy odor, has actually been a big concern in drinking water in recent years. So that's kind of interesting. We've got some methane oxidizers. We've got things that degrade 2,4-D, which is a pesticide, actually, which is kind of cool. And then we have nitrogen fixers, and then we actually have things that are pathogenogenic to plants, so they actually cause disease in plants. So these are some of the big ones that we care about. Last thing that we'll talk about is kind of distribution of microbes in soil. So they have high density and diversity. So there's up to 10 to the 8th culturable organisms per gram of dry soil. We're going to have bacteria, protozoa, algae, and fungi in there. So this is culturable organisms, not just bacteria, it's everything now. Normally, we have what we call a patch-like distribution of organisms due to colony formation. So in soils, we tend to see colony formation happen quite often because there's enough particles to latch onto and to work with. And then in surface soils, normally the distribution is also determined by texture and structure of the soil itself. So pore size is going to be really important for moisture content. So gram-positive bacteria have actually been found to be better adapted for dry conditions, and so they'll end up in the exterior of a soil aggregate. And gram-negative need more moisture, so they prefer the interior of the aggregate because it shields them and keeps the water near them, which is really, really interesting. There's a lot of morphology things that you can learn about soil that kind of tell you where those organisms like to live because of how they move water or retain water, which is kind of interesting. So this is just a little picture to kind of show what that looks like. So this is like a soil particle cross-section with different things that are being secreted in different colonies, just to give you a sense of kind of how complicated this is in the actual soil. And then for the most part, when we're talking about microbes in soil, about 80 to 90% of cells are absorbed to solid surfaces. So we're seeing that colony formation. And basically the purpose of that is that attachment is actually advantageous for bacteria. It protects them from predation. It helps to localize concentrations of nutrients really close to them within the aggregates. And it actually also helps to mediate pH. So if you're within a colony, you're kind of helping each other mediate that pH, mediate changes in moisture, and sharing nutrients sometimes. So the last thing that I want to leave you with at the end of this pre-lecture is how could you use how soil and soil microbes in particular are distributed to your advantage. So the fact that they're in colonies, the fact that they like pore size or don't like the pore size, or they're on the aggregate or in the aggregate, like what would that help if we're thinking about designing things as environmental engineers? So I'll leave you with that thought. Think a little bit about that and we'll discuss it in lecture.